All right, for our next example of how neurons can be functional or wired together to do something useful, will be in the realm of hearing. So what you see now is a diagram of our auditory system. So your outer ear will be over here. Here's your eardrum. And you might remember that we've got these three little bones in the middle ear. And so when the eardrum vibrates, those little bones move. And those little bones are hooked up to these little, another little window here in the inner ear. And the vibrations of this little window is going to set up vibrations of fluid in this inner ear is going to transmit the disturbance, and there's this little membrane that is going to vibrate, and we have little hair cells that are going to respond to the vibrations of this membrane by sending electrical signals to our brain, and that's what it is to hear something. So let's take a look. So here's the idea. When the eardrum, play that again. When the eardrum is moved by sound waves hitting the eardrum, it's going to move the bones in here, and then the oval window is going to deform a little bit. Now, the oval window is this top one that our three bones are connected to, but there is this round window down here that also deforms in the opposite way. And so what this is creating is movement in this basilar membrane. So the basilar membrane is where these hair cells are located, and they're going to be responding to this physical disturbance by sending electrical signals to the brain. Here are these hair cells, and they are connected to, ultimately, the eighth nerve, which is our auditory nerve, carrying sound information to our brain. Now, it turns out that these hair cells are located along the entire length of the basilar membrane. And this sets up a situation where these hair cells will be responding to different frequencies of sound. Now, the way this animation works here, the highest pitch is shown to be registered by hair cells closer to the oval window. Hair cells that are farther down the membrane are maximally responsive to lower frequency sounds. This is called place coding. So depending on where you are as a sensory neuron, you are carrying information about a different aspect of the environment. In other words, if you're a hair cell located close to the oval window, then you respond to high frequency sounds. If you're a hair cell down here, you respond to low frequency sounds. And so the placement along the basilar membrane is a sort of a map of the frequency of sounds that are being detected by the ear. So you can imagine then a pattern of activity of hair cells along this membrane would be what it is to hear some complex sound. So the frequency of the sound that you hear is coded, is represented by the activity of hair cells that are distributed in this on this membrane. And again, this is called place coding because certain frequencies are activating cells at a certain location on the basilar membrane, and other frequencies, the low frequencies, are detected by cells further along the basilar membrane. Now, what about loudness? So loudness, for any particular frequency, so notice what you're hearing is just the same tone, but the first one is loudest, then the second one is a little less, and the third one is softer. The loudness of the tone is registered or represented in a different way. It's not place coding anymore, but it is the frequency of action potentials. Unbelievable. So the loudness of the sound is not encoded by the placement of the cells. After all, it's the same hair cell that's responding to that same tone. But if it's a loud tone, the hair cell generates high frequency action potentials. If it's a soft tone of that same frequency, then the hair cell generates a lower frequency of action potentials. So that's called frequency coding, because it's the frequency of action potentials that is correlated to the loudness of the sound. So we've got place coding represents the frequency of sound, so the tone, and then frequency coding is representing the amplitude of the sound. Okay. So complex sounds, we can imagine, are activating the whole set of hair cells along the length of the basilar membrane, depending on which frequencies are present. And then the loudness of each of those frequencies is being represented by the frequency of action potentials generated by each of the hair cells. So let's just sort of review this quick second. Here's our situation: eardrum connected to the bones, and that's going to vibrate the oval window and the round window, and that's going to set up these vibrations in the basilar membrane. And we've got hair cells located along the length of the basilar membrane. Each of the hair cells can generate action potentials in a nerve, the eighth nerve, which would be sent to the brain. Place coding is the idea that each hair cell is responsive to a certain frequency. High frequency sounds activate the hair cells nearest to the window. Low frequency sounds activate hair cells farther away from the window. The loudness, or the amplitude of each frequency, is represented by the frequency of action potentials that are generated by each of the hair cells. Okay. Now, we're going to move on to another aspect of the auditory system, and that's the ability to localize a sound in space. And so if you've ever just been in a room and somebody starts talking, you can orient to where they are. Or if you hear a clap or some noise in the room, you can orient to where it came from. Well, that is a computation that neurons are doing. And so a fair amount is known about how brains are doing that. So to understand that, we're going to first have to take a look at the neural pathway for sound entering the brain. So here we have, a brain. So it's a coronal section of the brain, and the ears here. Sound would be hitting the outer ear, and so we're going to vibrate the eardrum. The little bones are going to make waves in the basilar membrane, and information will be sent up to the brain. If you take a look on the right here, here's the cochlea. And the auditory nerve then is right here. So that's the nerve carrying all the information from those hair cells. That's the auditory nerve. And it makes a synapse in a region of the brain called the cochlear nucleus. And then cells in the cochlear nucleus send axons to another region of the brain called the superior Oliveri nucleus. Cells in the superior Oliveri nucleus send axons up to a place called the inferior colliculus. And cells here send axons up to thalamus. And cells in the thalamus send axons up to auditory cortex in the temporal lobes. So there's a relay of neurons carrying sound information up to auditory cortex. Now, it turns out that in order to localize a sound in space, you need information from both ears. And the brain is going to make a computation based on inputs from both ears. And the first place in this system where information from both ears converges is the superior Oliveri nucleus, right here. And so now we're going to take a look at a magnification of the superior Oliveri nucleus. So here it is. Now the idea here is, again, here's the left ear and here's the right ear. Here are the hair cells, and when they vibrate, they send action potentials down. Here's the cochlear nucleus, that first relay station. And here we are, the superior Oliveri nucleus. Notice that sound information from the left ear and the right ear is converging in the superior Oliveri nucleus. So it's here that researchers suspect, and there's good evidence for this, that a computation is happening here. And what the system does is to detect the delay in sound information, action potentials reaching the superior Oliveri nucleus. The delay between the left and the right can be used to compute the location of the sound. And that's because when you hear your two ears, if the sound comes from this direction, well, the sound will hit this ear first and this ear a little bit later. So that delay, the amount of time it takes for the sound to get from here to here across the head, doesn't seem like much, but the nerve cells can detect that delay, and they can use that then to compute where the sound came from. If the sound comes from directly in front of you, it's going to hit both ears simultaneously. If the sound comes from over here, it'll hit this ear first, followed by a delay, and then this ear will register the sound. And so the superior Oliveri nucleus seems to be detecting the delays in the sounds as they arrive at both ears. So we're going to look for how it is that cells in this superior Oliveri nucleus are able to detect this temporal delay. That's the key. Well, when they looked at the arrangement of cells in the superior Oliveri nucleus, they found something like this. So these are cells in the superior Oliveri nucleus. We'll just call them 1, 2, 3, 4, 5. And here come the cochlear nerves from the left ear and the right ear. Now notice what we got here. We got 5 cells here, each receiving terminals from each of the cochlear nerves. But notice the right nerve, the red one here, has a long axon here to send information to cell 1, whereas the cochlear nerve on the left, the blue one here, that's a short axon. So now we can begin to see how activity of these neurons here, the green ones here, might be registering the delay between the sound arriving at left and right ear. Here's how the system would work. Let's imagine that the sound comes from directly in front of the person. So the right and left ear will be activated simultaneously. Now, action potentials then will be generated along each of these axons. And notice by the time the action potentials arrive here, the blue one here, the one on the left here, has already reached cell number 1. Now here are the rules for how the system operates. These cells will only fire action potentials of their own when they receive simultaneous activity from both of the inputs. So this would be an example of spatial summation, that the cells will only generate their own action potentials if they receive simultaneous input from both of the nerves here. So in our case, when the sound comes from the center, the action potentials get started off at the same time. By the time it reaches cell number 5, it reaches cell number 1 over here. But since there's not activity in the other terminal, cell number 1 and 5 will not respond. By the time this action potential gets to 4, this action potential gets to 2. But again, since there's no activity in the other terminal, these cells won't fire. By the time the red one gets to cell 3, the blue one has arrived at cell 3 at the same time. You get simultaneous activity of the inputs. That drives the cell to threshold. It generates action potentials. So cell number 3's activity, what that represents would be that the sound is coming from in front of the person, right? Cell 1, 2, 4, and 5 did not respond. So cell 3's activity is, you can think of it as representing a certain amount of time delay between the sound getting to the right and the left. In this case, the time delay was 0. Action potentials converge on cell 3 at the same time, and only cell 3 at the same time. Now imagine the sound comes from the left. So it's going to hit the left ear first, and the right ear sometime after. In fact, this would be the greatest amount of delay in time, right? So again, action potentials always travel at the same speed, and remember, action potentials in an axon that branches, they go down both paths. So here we go. The action potentials race off. Here it goes. We're going to cell 5 and cell 1. This one gets This one gets activated first. So this one has a head start. So this one, the action potentials might reach cell number 1 prior to the right ear even registering the sound. But since there's no activity here, because there's no action potentials being generated yet, cell number 1 does not respond. So the action potential continues along to cell number 2. But the sound hasn't even reached here yet, right? Then cell number 3. Now maybe the sound begins to reach this one here, cell number 4. There's no coincident activity. And finally, action potentials will meet at cell number 5. So cell number 5 is activated simultaneously by its inputs because the left nerve had a head start. The action potentials had a head start. Of course, they arrive here, but there's no corresponding activity in the red pathway, so the cell number 5 gets coincident activity, and it will reach threshold and generate action potentials. So when cell 5 is activated in this system, then that is effectively a computation that there was maximum delay and that the sound came from the left. And now you can just imagine, what if the sound came from the right? Well, then it would be cell number 1 that gets coincident activity between the two. Because again, if the sound comes from the right, these action potentials have a head start. By the time they get way down here, then the left ear response sends its action potentials to cell number 1, becomes responsive, and it then is computing that there is maximum delay, and so that means the sound came from the right. So cell number 1, if it's active, then the sound came from the right, 90 degrees to the right. If cell number 5 is active, the sound came from the left. If cell number 3 is activated, the sound came from either in front of you or behind you, right? So theoretically, cell 3 would be activated under both of those conditions. There are other ways to figure out if it's in front of you or behind you. Like it's a little bit louder if it's in front of you due to the way your outer ears are arranged. So if we now see this system as hooked up to motor systems, remember, if cell number 3 is activated, it's going to engage motor systems that will make you orient straight ahead. If cell number 5 is activated, it'll engage motor systems that will make you turn left. So your head movement is going to go left. And if cell number 1 was activated, it's going to engage motor neurons to make you turn right, because the sound came from the right. So sometimes this network is sometimes called, they're called delay lines. The way this system is arranged, the network of these neurons, it enables it to detect delay, the delay between activity of the two inputs. These cells then, depending on which one is activated, are in a sense measuring the time of delay. And since the time of delay varies with where the sound is coming from in the environment, this system can be used to localize sound.